My presentation for mammal doubly labeled water for today is the paper Energy Expenditure and Personality in Wild Chipmunks by Coreau et al. 2015. It was published in Behavioral Ecology and Sociobiology. So, what is the question this paper is trying to ask? Coreau et al. Wants to know if personality and individual behavior is related to energy expenditure in free-living organisms in their natural habitat. This study also asked if there was a link between resting metabolic rate, daily energy expenditure, and the personality traits of exploration and docility in wild eastern chipmunks. And we'll go more into what exploration and docility mean in a minute. These behaviors have been previously examined in laboratory studies with captive chipmunks, but the authors wanted to determine if the same analysis holds true in the field. Basically, they wanted to get a more accurate depiction of how behavior is reflected in energy expenditure in the natural environment. So why this question? The authors are examining the pace of life syndrome concept, otherwise known as POLS. POLS suggests that so-called fast-living species, that is, ones that grow quickly, mature early, and die young, have a higher base metabolic rate than slow species. Ones that mature later, breed later, and typically have a longer lifespan. POLS also suggests that the two are associated with opposite phenotypes. Previous POLS studies more or less neglected behavioral traits. So POLS is potentially applicable at different levels of organization, although there is little understanding of how it works at an individual level. The studies have largely neglected behavioral traits in the past, partially due to the difficulty in measuring behavior comparatively between species. One of the key predictions of POLS theory is that faster, more aggressive explorers should have a higher daily energy expenditure than less active, slower explorers. Previous studies supported that assumption by studying base, resting, and standard metabolic rates. However, as these studies mostly take place in laboratory settings, it is difficult to know if the results hold true in a more natural setting. To properly understand an animal's daily energy expenditure, we need to be able to observe the full range of complex behaviors that are exhibited in the field. So, why this species population? The species being studied here is the wild eastern chipmunk, T. striatus, which is a fast-living species under the POLS concept. And this is a population that the authors had studied in the past and are more familiar with. They found that the population displays consistent exploration patterns, ranging from fast and superficial exploration to slow and thorough. In this definition, fast explorers start with highly intensive exploration, substantially decreasing after a period of time. Slow explorers, on the other hand, exhibit moderate but constant exploration throughout. Fast explorers also exhibit low docility, higher trepidability, and lower hypothalamo-pituitary adrenal reactivity than slow explorers, and reproduce at a younger age. In other words, individual differences in exploration behavior are likely to have physiological implications which would be reflected in the measures of DEE. So, the study population is located in the Reuter Valley Land Trust of Sutton Mountains, Quebec. I tried to get a better picture than this, but it was a lot blurrier than I expected. Reuter Valley Land Trust has a lot of oak, beech, and maple trees that are relatively mature, which means there's abundant mast for the chipmunk population that will help them survive throughout the year. This is important because the study itself takes place year-round. It's cut up into three periods throughout the year, 4 months, every 4 months. And the study itself went on for quite a long period of time, about 5 years. So having a site with an abundant food source meant that the population was more stable and more easily studied. To measure DE, the chipmunks were live captured, tagged, and injected with 240 microliters of doubly labeled water. Toenail clippings were taken as an initial blood sample, and they were held for an hour to equilibrate and released at the original trap site. After 1 to 3 days, the chipmunks were recaptured and weighed, and another blood sample was taken. This was done as close to a 24-hour window as possible. As we've learned from previous lectures, DLW is most effective within a 24-hour period of recapturing. Resting metabolic rate, or RMR, was measured using a computerized open-circuit respirometry system. The system provided a constant flow of 450 milliliters per minute of air into each chamber. So this is a 2-chambered system. It's difficult to find a picture of that as well, so you'll have to use your imagination for this. Chipmunks were observed overnight using measurements of oxygen consumption and visual confirmation to determine when the chipmunks were at rest. They basically just read the computer analysis and watched to make sure that the chipmunks were not moving. To avoid unnecessary pressure on chipmunk survival, the specimens were captured in the late afternoon and observed overnight. That way, they would be observed during their natural sleep cycle and would not miss on foraging or mating opportunities. Chipmunks were also provided with unlimited peanut butter and apples, so they could be measured at optimal energy intake. Basically, they wouldn't have to worry about post-absorptive energy consumption factoring into it. So the researchers wanted to make sure that as much energy was available as possible. To test docility, they used something called the handling bag test. Chipmunks were placed into a mesh handling bag and suspended for 1 minute. Docility was measured as the number of seconds each chipmunk spent immobilized in the bag. The study was intended to mimic freezing behavior in the wild and has been used in previous studies to test an individual's behavior in response to humans. Open field exploration was measured using exploratory arenas of different dimensions, which had a gridded floor. So the arenas consisted of 1 or 2 white plastic boxes of 2 sizes. The small arena was 80 by 40 by 40 centimeters. Large arena was 100 by 100 by 50 centimeters. Dimensions aren't necessarily important here. The important thing to remember is that there is a gridded floor of 3 by 6 grids. Level of exploration was measured as the number of times an individual crossed over a grid line during a 3-minute period, with high scores defined as fast individuals and low scores as slow individuals. To minimize variation in these definitions between slow and fast individuals, all analyses of open field exploration were done by a single person. Basically, they didn't want an inconsistent measurement of what consisted a fast versus a slow explorer. Data was analyzed using a Markov chain Monte Carlo multivariate mixed model. I don't want to go too much into the math. I didn't completely understand it myself, and it doesn't seem particularly relevant to the rest of the paper to go into a lot of detail, so I'm just going to briefly touch on it and just say that researchers estimated correlations between metabolic and personality traits on an among individual level. The data was run through a random effect variance of individuals model 6.5 million times, cutting the sample size down by 1.5 million each time. No, 150,000. To avoid influence by intrinsic and extrinsic variables, researchers fitted fixed effects of age, sex, and reproductive status for all variables based on previous studies. basically to ignore any unnecessary confusion from those data points. Though age, sex, and reproductive status could all have their own influence on the DEE and personality traits. So using a fixed measure basically just takes them out of the equation. So what did they find? The major... Sorry about that. The major results for the study are compiled in Table 2 here. The multivariate mixed model showed significant variation in all 4 variables among individuals. I would like to note here under where it says trait v individual, which refers to the among individual variance, you can see a rather significant variance for each of the methods being studied. But note that variability in resting metabolic rate was less than half of any of the other traits. More of the findings are provided here on Table 3 and Figure 1. In Table 3, they use a measure called the 95% highest posterior density, otherwise known as HPD. It is used as a measure of confidence, similar to the 95% confidence interval that a lot of you are probably familiar with. An HPD interval that overlaps with 0 is not considered to differ significantly from 0 and indicates a lack of relatedness between the 2 points. Note that between the lower and upper HPD intervals listed in Table 3, the factors that are significantly correlated are DEE with exploration and exploration with docility. This is visually represented in Figure 1 on the left. DEE and docility are not shown to be significantly correlated. It should be noted that docility and exploration are negatively related and relatively well bunched together. It's a little difficult to tell based on this image, mostly because the outlier creates a problem, but DE and exploration are relatively well linked. They used a confidence interval here instead of the HPD interval, which could result in different results. All right, so what do these findings mean? Negative correlation between daily energy expenditure and open field exploration could indicate that environmental effects impact daily energy expenditure. Alternatively, there could be a genetic component between DEE and exploratory behavior. It is important to note that lab-only studies did not find a correlation between exploration and docility, as this one did. Previous studies did focus on phenotypic analysis instead of behavioral traits, which may have been unable to detect a pattern between the two behaviors. That this study does indicate a correlation between the two indicates that environmental influences are important in studying personality traits, and lab-only studies may not be enough. Finally, a major finding of the study is that fast explorers have lower DEE than slower explorers, regardless of docility. This is counterintuitive in the PLOS theory, which indicates that faster-moving individuals should have a greater energy expenditure. One explanation of this finding could be the relatively short open field measurement compared to other studies. You may recall from the beginning of the presentation that slow explorers retain a constant level of exploration, while fast explorers start at high levels of exploration, dropping off after a period of time. Over time, it is possible that slow individuals would explore more in total than the faster explorers. If the open field study had lasted longer than 3 minutes, it is possible that we would have seen more line crossing on the slow explorers than on the faster ones, which would support the pace of life syndrome hypothesis. Basically, the fast explorers are not exploring. After 3 minutes, the fast explorers aren't exploring much at all, while the slow explorers are still exploring at the exact same amount. All right, so conclusions. The message I would like you all to take home from this study is that personality traits have an impact on energy expenditure in natural environments. For future projects, more empirical data is needed to show that open field tests really do reflect natural environments, like the study predicted it would. The author suggests studying other species in a similar manner to be completely sure that natural environments are actually mimicked in an open field. For this population, the next area of study would be how to relate resting metabolic rate and daily energy expenditure to measures of activity in their natural habitat and their life history strategies.